So what I'd like to do is just basically run through the steps involved in how signals get sent through neurons, from neuron to neuron, and then finally to muscle. Right? This is basically going to sum up the steps involved in what we looked at with neurophysiology and the muscle physiology. Now, there are a few parts we haven't seen yet. Right? So you can imagine for all of this to occur, this is going to be about how do we produce a response. But the response has to be coming from somewhere, usually from some sort of signal. In Chapter 13, we're going to go into the places where those signals come in. Right? They're called receptors, and there are a whole bunch of different kinds of receptors. Okay? Receptors can sense what's going on outside of the body. Those are going to be called exteroceptors, or inside the body, those will be interoceptors, and we'll see those all more in depth in Chapter 12 and 13. Some of these receptors are specialized neurons, and others are cells that talk to neurons or send signals to neurons. Okay? So let's just assume for a moment that a signal has been received by one of these specialized receptors. Okay? What that signal is going to do is going to cause what's called an excitatory postsynaptic potential, right? A change in potential in a neuron, and that neuron will eventually transmit an action potential. So what we see is an action potential coming in from this preliminary neuron into the neuron that we'll be focusing on. Right? This is an example of a multipolar motor neuron. Right? We see multiple dendrites coming off. As this action potential arrives at the dendrite, here we have a synapse between the axon terminal of our preliminary neuron and the dendrite of our multipolar motor neuron. Here we're going to see this action potential signal changing into a different kind of signal. It's going to change into a chemical signal. Right? In this case, if we were to focus in on this synapse here, we would see that the axon terminal is going to be the site of neurotransmitter release. Those neurotransmitters will be released, and they will bind to chemical receptors that also are ion channels. When this neurotransmitter is released, it exocytoses and binds to its channel. Those channels will open and will lead to some sodium ion coming in. Now, of course, this assumes that this dendrite, and in fact this entire neuron, begins at resting membrane potential. So that resting membrane potential, some of the key things to remember about it, it is measured at minus 70 millivolts, meaning that the inside is 70 less relative to the outside of the plasma membrane. That we have a greater concentration of sodium ion outside the plasma membrane and a greater concentration of potassium ion inside. Right? Those are the basic steps that we need always when we talk about resting membrane potential. What will occur when we open this channel and sodium starts coming in, we will start bringing some of those positive ions from the outside in. And that's going to lead to a change in this resting membrane potential, and we call that a depolarization. Now, these depolarizations are fairly small. They are localized to the area where the binding is actually occurring, and the further from the binding, they become weaker and weaker. Okay, so we have what's called transmission with decrement. These then, occurring in the dendrites and also in the cell body, are the graded potentials. So we have graded potentials occurring where we have this preliminary neuron, its axon terminal, talking to the dendrites. Now, again, we're not going to see a single neuron talking to a single neuron, so we can assume there will be many neurons communicating with this motor neuron, or we'll see many signals coming in from this preliminary neuron. What occurs with these graded potentials is these can sum up either over time or over space. Right? We call that temporal or spatial summation. And the goal with these graded potentials is to either initiate or not initiate an action potential. What's required, if we focus on this area right here called the axon hillock, this axon hillock is rich with voltage-gated sodium channels. And these voltage-gated sodium channels are where we will see the beginning of action potential occurring. These voltage-gated channels open with a certain change in membrane potential. Okay? We call that threshold. The threshold that we will use, we focus on, is minus 55 millivolts. That means the actual size or magnitude of these graded potentials have to be able to take the resting membrane potential from minus 70 millivolts at rest to minus 55 millivolts for threshold. When this occurs, right, so if we have an actual magnitude of graded potential equal to or greater than 15 millivolts, the difference between minus 70 and minus 55, that will cause the voltage-gated sodium channels here in the axon terminal to open. In the axon hillock to open. When we open voltage-gated sodium channels located here in the axon hillock, this will lead to a very rapid and very large depolarization. That depolarization, that is going to, if you imagine the depolarization from one channel, will lead to being above or super threshold for the next channel. That channel will have sodium rushing in. That sodium rushing in will lead to depolarization, opening the next channel, and so on and so forth. Once we have started action potential here in the axon hillock, it will continue the length of the axon without changing its magnitude. So the way in action potential, if we look at any point, we'll just focus on this random point on the axon. When the action potential is arriving there, right, it arrives as a depolarization that is super threshold. It is above the minus 55 millivolts. That occurs because these voltage-gated sodium channels have opened. Sodium will move in. That sodium is going to be above threshold, so we'll open the next voltage-gated sodium channel. So within our area that we're focusing on, we'll have a rapid depolarization. We can visualize this with a trace, with time in milliseconds on the x-axis, and the membrane potential measured in millivolts on the y-axis. At rest, we have minus 70 millivolts. Threshold is minus 55 millivolts. Okay. In the axon hillock, this region, right, this initial depolarization, would be due to the previous graded potentials. Once we are talking about the axon itself, this depolarization would be due to the depolarization caused by the previous segment of the axon. At minus 55 millivolts, these voltage-gated sodium channels open, and we have a very rapid depolarization, including a reversal of membrane potential, so that the membrane potential actually switches and becomes positive, meaning that now the inside is more positive compared to the outside. That goes up to plus 30 millivolts. And at plus 30 millivolts, two very important things happen. One thing that happens is these voltage-gated sodium channels will close, stopping the influx of sodium. The other thing that will happen is we also have voltage-gated potassium channels. Those voltage-gated potassium channels will open. But potassium is at a higher concentration inside of the plasma membrane, so the potassium is going to start flowing out. Right? When that occurs, we will see a rapid repolarization. Okay? So that repolarization is due to potassium leaving. Now, more potassium leaves than is really necessary to bring us back down to minus 70 millivolts, and that's because those potassium channels, they tend to open slower and then close slower. So we get an extra amount of potassium leaving. That brings us down a little below minus 70 millivolts into what's called hyperpolarization. That hyperpolarization is really important because that's going to be what keeps these action potentials moving in only one direction. When the potassium voltage-gated channels do finally close again, resting potential is restored mostly by the sodium-potassium pump, which utilizes ATP to pump sodium out and pump potassium in, bringing us back to those resting membrane conditions of, again, minus 70 millivolts difference, increased or higher concentration of sodium outside, and higher potassium inside. Okay, once again, that is rest. You must always think about action potentials, graded potentials, as changes from resting membrane potential. So that is what would occur at any point. Right? When we look at how this signal flows in only one direction, the reason it flows in only one direction is while the cell while the area or the segment of the axon is actively within the depolarization phase or in the repolarization hyperpolarization phase, it cannot undergo another action potential. So that's going to keep this action potential moving in the forward direction. Now this would be continuous in short unmyelinated axons. However, some very long axons have a myelin sheath. And what that myelin sheath does, right, if we think about the segments of myelin sheath made of those Schwann cells, the action potential occurs in this segment, and then the depolarization is insulated so it slides through. Then we have action potential in this segment, slides through. Okay. That is going to increase the speed, and this is really necessary when we're talking about a very long axon. Right? Transmission through action potential in an unmyelinated axon travels about 2 meters per second, which seems kind of fast, right? But if you take an average male human who is about 2 meters tall, it takes a second for a signal to travel the length of that individual. That's not very fast. Myelination can actually make those signals travel about 100 meters per second, so 50 times faster. Now, switching to the other side of things, right, once this action potential has successfully made its way through a neuron, it may end up at a muscle finally. In this case, it does end up at a muscle because we're talking about a motor neuron, and motor neurons terminate at neuromuscular junctions. Right? Neuromuscular junctions is when the signal from the neuron is going to speak to a muscle or transmit to a muscle to lead to muscle contraction. So if we want to focus in on that neuromuscular junction, right, what we have roughly sketched here, this would be the axon terminal of our motor neuron. This is the motor end plate of the muscle cell. When this action potential arrives here, it is going to cause calcium to come in through calcium voltage sensitive channels. That calcium is going to take some synaptic vesicles that are full of neurotransmitter. In the case of neuromuscular junction, it's actually acetylcholine. And is going to move that acetylcholine-filled vesicle to the plasma membrane for it to exocytose, and the acetylcholine will pass out. When the acetylcholine is exocytosed, it will bind to a ligand-gated channel. Its receptor is a ligand-gated channel. That channel is going to open, and sodium is going to come in, leading to a depolarization. This depolarization is the same thing as the graded potential we saw previously. If there is enough of these graded potentials, because again, I'm drawing a single molecule of acetylcholine here, please understand, you don't see a single molecule being released. If there is enough depolarization occurring here in the motor end plate, that will kick off or induce an action potential in the sarcolemma, which is the plasma membrane of the muscle cell. That action potential will travel along the sarcolemma and pass down through the T-tubules. Right? When that action potential passes down through the T-tubules, it is going to come in contact with the voltage-sensitive calcium channels, releasing calcium ion from the sarcoplasmic reticulum. This is going to be the second job that we've seen of calcium in this. Calcium did one job on the neuro side of things and another job on the muscle side of things. So what's going to happen with this calcium then? That calcium is going to bind with the regulatory subunit in the sarcomere, that is troponin. That troponin will change shape and will pull tropomyosin back. When tropomyosin moves away from actin, the actin binding sites are exposed. Myosin can then interact with actin. When we have a rough sketch of our sarcomeres, when this myosin can interact with actin and there's ATP present, that myosin, those myosin heads will ratchet inward, bending inward, and will contract, shortening the distance within the sarcomere. And when all of the sarcomeres contract at the same time, every myofibril within a muscle cell will contract. And when every myofibril contracts at the same time, because remember this neuron is talking to the entirety of the muscle using action potential, which travels really quickly through the T-tubules and through the sarcoplasmic reticulum, we get contraction of the muscle. Now when I drew this, I only drew one axon terminal talking to one muscle, but please remember we can have many axon terminals talking to different parts of the muscle, right? Depending on how many neurons talk to how many different cells within the muscle. That would be our motor unit. So when we have fine dexterity control, we have very small motor units. Okay? When we have gross movement, something like contraction of the biceps, we have much larger motor units. So this is how this all fits together. So chapter 9 and chapter 11, the information on the physiology part isn't really separate. Right? When you think about it all together, it's all action potential. If you understand the steps involved in action potential here in the neuron, it will be similar to what you see in the muscle cell. The idea of graded potentials, right? The idea of how we go from an electrical signal, right, an actual change in membrane potential, to a chemical signal that can cross a synapse, that can cross a space. Right? Those are basically the main concepts that you need to remember and put together when you're talking about the chapter 9 and the chapter 11 physiology portions.